So this is originally a 30-minute talk, and so now it's going to be sort of a whirlwind, really. There's a lot of data. A lot of this has been analyzed in the last year. You guys have seen sort of bits and pieces of this at previous conferences if you've been in Orlando or Silver Springs. But now we've sort of come quite a bit ways, and I showed you also some of this at the family conference, so now I'm sort of excited to share this with you today. So these are the easy parts to sort of skip through, just to show that in our lab, in collaboration with Stormy Chamberlain, are studying induced pluripotent stem cell-derived neurons from patients with Angelman syndrome and Dup15q. And as you saw yesterday, Eric had sort of talked about our Angelman stuff, our big Angelman story, which was published earlier this year, and then some of the sort of early parts of the fellowship looking at the development of the Dup15q iPS-derived neurons. And as I just mentioned, we're just using induced pluripotent stem cell-derived neurons to model this, and these are just neurons that are derived from somatic cells from patients through some magic that gets turned into stem cells, and then those become neurons. And as we had a large discussion yesterday, these are neurons from the patients, so they had the same genetic changes that the patients have. And so this is just our timeline, just to give you a feel for how we do these things. The stuff that Eric talked about yesterday is this sort of early part of Angelman, which is this development, because these are neurodevelopmental disorders. So you're really interested in looking how these things change over time, plus activity-dependent plasticity, which we know is really important in Angelman syndrome through Dr. Philpott and other people. And then also he showed the Dup15q week-by-week development, because we thought when we saw the Angelman phenotype that this might be true for all neurodevelopmental disorders. If you have impaired intrinsic properties that are not maturing the right way, maybe that's just non-specific, right? And we're talking about biomarkers and things that are specific to Dup15q. So that's sort of the logical step. And as you saw yesterday, there's not many major changes with the Dup15q development. So the second part is this sort of higher order. I like to think of it of like sort of later, more mature processes that we looked at these sort of late ages where no one at this point has recorded from an iPS-derived neuron. So these are beyond 20 weeks in culture, so it takes a long time to get to this point. But at this point, they start acting much more like neurons you would expect neurons to act. So they have more plasticity. All their features are, as Eric mentioned yesterday, almost 100% much more mature than they are at these early stages. So here's my recap slide of Eric's talk yesterday. It's just that Angelman neurons have this impaired development across multiple features. Just some suggestive evidence that we did with the potassium chloride says that the resting memory potential might be primary. And I just want to repeat this because this may also apply to this Dup15q story I'm going to share with you today. And that the same types of differences are not true for Dup15q, and except for the plasticity. So that seems also to be disrupted. So this suggests a distinct mechanism for hyperexcitability in Dup15q, right? So there's one for Angelman syndrome that does not apply to Dup15q, and the stuff I'll show you today is also true. The stuff for Dup15q is not true for the Angelman syndrome cells. So we have a bunch of different lines. Today I'm going to show you, in most cases, data from, I think, 9 control subjects, a paternal duplication individual, and then 3 IDIC patients plus 1 interstitial triplication, as well as 3 Angelman subjects, 2 of which are large deletion and 1 has a mutation to UBE3A. So keep in mind, blue is control, green is the paternal duplication, purple the Dup15q, and Angelman are in red. So to study these, we're using electrophysiology. So we just take a very sharp glass pipette attached to an electrode, bring it to the membrane of the cell, apply some suction, and then you can start sort of listening into the conversation between neurons. So this is some slides I showed at the family conference, but we're studying pretty much synapses, which are just the connections between cells, and seeing how these neurons talk to each other. So there are a few things you need to keep in mind for the rest of this data. Is there's 2 types of activity. Action potentials, which use large electrical signals, you can think them as, or synaptic activity, which are sort of this smaller chatter. So now getting into the data, this is sort of what the fellowship was initially based on. It was that when we looked at control subjects, here on top we're looking at frequency of the action potentials, number of these electrical signals over time, so events per second. And here are 9 different controls. In purple are Dup15q neurons, again, paternal Dup and Angelman, and you can see that across the controls, the paternal Dup and Angelman, they fire an intermediate amount of action potentials, right? So about half a hertz activity. But the Dup15q neurons, in all 4 cases, fire double and often triple to some of the individual control subjects. So this is a lot of action potentials that are getting fired by Dup neurons compared to controls. That's not just because of one patient, right? This is across all 4 here. The paternal dupe is just all the other, the GABA receptor subunits are duplicated. They're not the same dosage as the IDEX because there's 3 copies in some cases, but this does get maybe some mechanism of is UBE3A playing a role, are other genes playing a role. And here, just to show you how specific this is to frequency, this is the amplitude of those electrical signals, and that's not different at all across any of these lines. And you can just see the group data here. These are all the lines combined. Controls, dupe go way, way up. Angelman and paternal dupe don't look any different than controls, and the amplitude is not different. We also can use calcium imaging to look at this. Now, instead of patching individual neurons, which takes a lot of time, we just look at many, many neurons at once by giving them a dye that lets them light up when they fire action potentials. And so here's a brief movie. Here's some low level. You can see that when one lights up, many other ones light up as well. And so if you look at this, for multiple examples I'll show you, this is one experiment that I pulled out from a recent experiment I did. You can see here controls. They have a bunch of these spikes, which are potentially equivalent to action potential firing. And in the dupes, the spikes are synchronous, which relates potentially to some type of network activity, something tying them together that's making them all fire at the same time, which may relate to the stuff I showed a couple slides ago. Here's another slide from a totally different experiment done a year or 2 before. Again, from dupes, you can see each of these spikes lays on top of one another, so again, very synchronous. And here's some more data. This is a roster plot, so each of these rows is a different cell from 2 control experiments from 2 individuals and 2 Dup15q experiments. And again, in controls, some fire at the same time, but many are not firing. But in the 2 different Dup15q individuals and 2 different examples in the last couple slides, again, all synchronous. Now, the next question was to find out what channel or receptor might be to blame for this increased activity or hyperexcitability. So we tried a few different pharmacological blockers on our cells to see if dupe neurons would respond differently than control neurons. And so again, purple's dupe, blue's control. Here's some traces of before and after, and this is here percent responders, the number of cells that responded to the drug. And you can see that for the most part, the dupes and controls aren't very different when you look at these are actually SK channels, blocking it, activating it. Here's the sort of non-specific SK. These are potassium channels. This is a T-type calcium channel, which some people are interested in with epilepsy and autism. That doesn't seem to be really different. You can see here this sort of decrease in firing. Here's some more drugs. This is interesting and sort of stuff we talked about today. If you block GABA, there's not much of a difference here. If you block inhibition, both controls and dupes respond with the same amount of increase in action potentials. That's not potentially related to the increases in GABA receptor subunits. So in this case, they're both responding the same to blockers of inhibition. This is another type of potassium channel. It doesn't seem to be different. But this channel here, I want to call your attention to. This blocker is for a channel called KCNQ2, which is related to benign familial neonatal convulsions. So these are kids who are born with seizures because of a mutation to this particular channel that goes away with time. And you can see that there's a significant difference here, here, and here between the controls and dupes. And in relation to decreased UBE3A and increased UBE3A, you can see that there's opposite responses between Angelman and control. So Angelman have a larger response and dupes have a dampened response. So this is sort of interesting. And the thing about KCNQ2 is it acts as a brake. So when you activate it, you press on it, you can reduce firing. But if you have a loss of KCNQ2 or something wrong with the brake, the cells will fire a lot, which potentially could explain that difference we see in the dupe neurons. So here is just group data of activating controls. You press on the brake, it goes down. If you block the brake here, it goes way up and looks much like the dupes at baseline. And you can see the dupes, when you press on the brake or block the brake, don't really change. The paternal duplication looks like controls, down, up. And Angelman actually has, I showed you his last data, which is very different, has an exaggerated response to these blockers. So they actually go up even more than the controls do. Here's the group data. So if you combine all the controls and the Angelmans and dupes, you get this nice significant effect with the activator and blockers and the controls. Same effect here on the Angelman. You can see the blocker works even better. And in the dupes, there's no significant change. And the amplitudes are not affected by the drugs. So then now we can look with the electrophysiology if the brake isn't working, the question is it because the brake isn't there? Is it there but not the same levels? Is there something wrong with it? So here's just some staining in red for the channel, this brake. Green are neuron markers. And then we quantified this using a few different methods, but this is just to say how much of the red is on top of the green, meaning how much of this channel is with the neuron, because it's expressed elsewhere. We can see in 3 different ways of quantifying these images from a couple different patients, the controls have a higher correlation of the red on the green than the Dup15q neurons have. Now, when you do this method of sort of staining for these channels, you're sort of screening for areas you can actually get a good signal, areas that have neurons. I mean, it's not as unbiased as other methods. So second thing we did was to separate these out by flow cytometry. So these are singleizing cells and then passing them through a laser, which can also read the fluorescence on each cell. But in this case, you're not sort of picking areas to take pictures. In this case, we quantified this, and we see that the percent of cells that have the brake is higher in both controls with 2 different neuronal markers than it is in the dupes. So not only is there less in the dupes based on the staining, but there's also less cells that have it based on the flow. So we have the electrophysiology that says they don't have as good of a response. We have the staining in the flow that suggests that it's down and maybe less cells have it. And now I want to turn to this sort of unrelated but seems to be related mechanism of synaptic activity. So I talked about this at the family conference, but if you go to synaptic activity, the experiment I'm about to show you is equivalent to sort of turning the music really loud, and then in response to that, you sort of turn the music back down, right? So this is called homeostatic plasticity. Here's the example I gave from a Neil Young concert, where these large amplifiers make the music really loud. Maybe the neuron's not very happy. You turn it back down, smaller amplifier, things are great. So here's an example from a review that looks like my data. So the blue is normal conditions. Here's the normal volume of the cell, let's say, of the activity. In green is when the volume goes less. So the music's been turned up, and now the neuron turns it back down. So all of a sudden, the amplitude just gets smaller. And if you actually hear, I think of this as if when you put earplugs in, and to hear the music, you turn it all the way back up again to compensate for the earplugs. Now when you take the earplugs out, the music's way too loud, right? So that's this yellow bar here. So to first measure this, this is looking at sort of receptors at the synapses called AMPA receptors. So these are ones that are in the membrane. And we use a vehicle, so this is pretty much nothing. Then blocking GABA, so this is going to block activity, so you're going to actually turn the volume up. You're blocking inhibition, so activity goes up, so you're going to turn the volume way down. And then TTX, which stops activity, it's the earplugs, which are going to make you turn the volume way up. And so if we look at the number of receptors with these treatments, we see here's vehicle. When you boost activity by blocking inhibition, you turn the volume down in green. And then red, when you block activity, you turn the volume up, which is what you expect. The dupes do not do this. Again, this is staining. If you go to the physiology, now we're looking at the distribution of these volumes for all the cells and events that we looked at. So blue is the normal conditions. Green dotted line here is when you turn the volume down. It's shifted to the left in control, just like it was in that picture I showed you. And the red's there, but in this case you can't see it because it's perfectly overlaid on top of the blue, which is not what we'd expect. We expect the controls to be able to turn the volume up. The Angelman can also turn it down, but can also not turn it up. And the dupes don't turn it down or up. So they have this impairment in turning the volume down. Now, this is sort of confusing because the staining says that the control neurons and the Angelman neurons can turn it up, the dupes cannot. And so we wondered that maybe the activity level is just not high enough in some controls to see this, meaning it's not active enough for them to want to turn the volume up when you stop it. So if we looked at the cells that had the most events, ones that are most active, and then looked at the ones that were blocked to see if they could turn it up. Now, in both these cases, so these are cells that had 4 Hertz activity, we see that both of them have this red line that's shifted to the right. So here, when you look at the best controls, you can see them. None of the dupes do this. So dupes cannot turn the volume down or up, and they have a lot of this exponential firing. The second thing I point out is this is a sort of different experiment, but here's just looking at the vehicle distribution, so the volume, the volume of the events between controls, Angelman, and dupes at baseline. And Eric showed this yesterday in a different set of experiments, but it looks like both dupes and Angelman neurons have this larger volume events compared to controls, even in these later experiments. The last thing is when you actually sort of wash these drugs away and see how they fire afterwards, you get this increase in activity. So here's what the controls look like. This is a totally different group of cells, totally different experiment. Controls, again, have this really nice response. The dupes have a dampened response. So this is just like the firing. This is 3 different ways for sort of the same experiment to look at this mechanism, and it seems to be disrupted in Dup15q neurons. So I've shown you that the Dup15q patient neurons are hyperexcitable. This hyperexcitability is due to an impairment in the brake, which seems to have cells have less of the brake, and potentially some cells just don't have the brake at all, and that they also can't turn the volume down. So this sort of hand in hand, not being able to adjust the volume plus already an increase in baseline might be sort of cellular version of seizure activity. There's a lot, I showed you in some of the movies, of bursting synchronous activity in these Dup15q neurons. So the next step is can we use pharmacological agents to normalize the brake? Is there something we can use to overcome this brake impairment that could then normalize the firing of Dup15q neurons? That's the ultimate goal. And the second thing is, can this cellular phenotype be seen in mouse models, and then can it be tied to seizures and behavior, right? Because seizures in a dish are not seizures. So I can say here all day, all this flashing and activity could be related to seizures, but until we can see a similar thing in the mouse, we can translate it, then we could say this is for sure tied to some type of behavior. So here's the working model, just that this hyperexcitability, and there's impaired volume, turning the volume up and down, and we're actually thinking that maybe this here can cause this, just like the resting memory potential differences in Angelman. So maybe blocking this in controls, we can mimic the inability for the dupe neurons to turn the volumes up or down. So with that, I'll just thank the Levine Lab and Chamberlain Lab, and especially the Dup15q Alliance for supporting this fellowship. And thank you.